Hi, my name's Elisa, and I'm a student at Carnegie Mellon University. If you're following our video tutorials, you should have just watched a video on plasmids, also known as vectors. In this video, we will be discussing bacterial transformation. We will be showing an actual protocol, as well as explaining the molecular biology behind each step of a bacterial transformation lab. We hope you enjoy.
To begin, you must understand what exactly genetic transformation is. Genetic transformation occurs when a host organism takes in foreign DNA and expresses a foreign gene. There are many steps in this process, but before you begin the transformation, you must have competent bacterial cells that will take in the foreign DNA. Competent cells are cells that can survive altering their cell walls to take in foreign DNA. These cells are usually still growing and are in the log phase of cell growth. There are a couple different ways to make competent cells. One method is electroporation, in which an electric current is passed through the cells, but the most common method is the heat shock method. In this method, cells can be made competent by suspending them in a solution of ice-cold calcium cations, followed by a brief heat shock, then incubation at 42 degrees, their most favorable temperature. Cells that are actively growing turn out to be the most effective transformants, also known as the best competent cells. The most common transformation bacterial cells used are E. coli cells. Now we will begin explaining the actual protocol. At 0 degrees Celsius, most likely in an ice bucket, the DNA is introduced to the competent cells. The DNA is gently pipetted onto the cells. You must be gentle in order to not destroy the DNA. Then the cells and DNA are allowed to sit. The DNA molecules and the phospholipids of the bacterial cell membranes are both negatively charged. Electrostatic forces are therefore likely to prevent entry of the DNA into the cells. However, the divalent cations, meaning a molecule or ion with two valence electrons with positive charges present in the cell suspension, may interact with the negatively charged phospholipids, coating the cell pores. Channels into the cells are thereby created, lined with positive charges, which attract the negatively charged DNA molecules. Some scientists believe the brief heat shock creates a thermal imbalance between the inside of the cell and its external environment, which therefore helps to pump the DNA into the cell. After this brief heat shock, the cells will be somewhat unhappy. In order to ensure that colonies will grow, you must add some sock to the tubes and agitate and incubate them. Sock is a nutrient broth that will be eaten by the cells and help them regain their structure and strength after the heat shock. Directly after the heat shock, the cells are put back into the ice bucket and allowed to sit for exactly 20 minutes. The cells are then placed into an agitator.
After agitation, the autoclaved agar plates are prepared and labeled. These agar plates contain the correct antibiotic that will kill off the bacterial colonies that did not take in the foreign DNA with the selectable marker. The selectable marker is like a survival gene. The most common marker is the AMP-R gene. This gene is resistant against ampicillin, which is the most common antibiotic used in agar plates.
Plate the cells carefully. Make sure to have a control plate of the transformed cells without the foreign DNA, labeled No DNA Control, as a basis of comparison. Between each plating, you must soak the hockey stick pipe in ethanol and then flame it to ensure that there is no contamination. It is also important to plate different amount of cells. For example, in one plate 50 microliters, in another, plate 200 microliters. This method will ensure that you have enough colonies or a few enough colonies to pick out a single colony. After plating, cells are then incubated overnight at 37 degrees Celsius. This allows them to recover from the treatment, to make DNA, RNA, and proteins, and to divide. The next day, you will be able to observe the colonies that have grown on each of the agar plates. This concludes the entire bacterial transformation protocol. The main concept to understand is during this process, bacterial transformations are taking in foreign DNA with a specific gene and a survival gene through pores created in their cell walls. If done successfully, the cells, when plated, will grow and express both the survival and specific gene. You must grow the colonies and isolate a single colony with the DNA from the bacteria.
We hope you now have a better understanding of bacterial transformation. If you continue with our video sequence, our next video will be on isolating genes and DNA in your newly transformed cells. We hope you enjoy and go have fun in Biolab.